All right, great. So I'm going to try to summarize some concrete proposals that were discussed over the last 2 days. I'm going to start with some ideas for data sets enabling variant discovery and effect prediction. The way I sort of designed this is try to define a problem, a proposal, and sort of approximating a data set. So the first sort of problem is that variant effect prediction models need to be trained on perturbation data. So this was, I think, this idea had many different flavors and had pretty large support from most of the folks here, that we designed some kind of large-scale mutagenesis experiment on approximately 10% of the genome, regulatory and transcribed, and measure multiple molecular profiles, cellular phenotypes, potentially kinetics, and in multiple cellular contexts and conditions. The challenge was really combinatorial explosion, and this is where I think ideas of how you design, how you have minimalistic designs, followed by potentially molecular profile imputation approaches to fill in the blanks. And there were some nice ideas on using extremes of distributions to design initial data sets and then sort of fill inwards. So that's one problem and proposal. The second is, I think there was a general consensus on the lack of appropriate data sets to learn causal trans-regulatory approaches, networks and pathways. And there were two proposals here. One was genetic, leveraging large data sets of natural variation to actually learn trans-QTLs. And the other was molecular, where the idea was to explicitly perform perturbations. Slightly different from the previous proposal, one way to converge would be to potentially focus on transcription factors. So the ideas would be to mutate, delete, inhibit, activate, change concentrations of TFs, and have multiple measurements, molecular measurements, do the same thing for regulatory landscapes of TFs, and do this in multiple cellular contexts, which would satisfy some of the cellular G by E requirements, various spatial and temporal contexts, time series, using microscopy for subcellular localization, and combinatorial perturbations, again, prioritized using physical interaction models, evolutionary constraint and selection, and feedback from computational models. So those were sort of two broad ideas that I think had broad support. Now going on to transforming predictive modeling paradigms, I think one very central area that needs a lot of focus is models that can generalize across contexts. There are a few examples here. We discussed PRS scores that change across population structure, genome architecture changes across cellular contexts, individual disease and species, variant effects change across measurement technologies and phenotypes. And so the proposal here is reasonably concrete, and of course you can riff on this in many different ways. But the idea is to learn a transferable, to create a data set and then challenge computational models to learn transferable models of molecular effects of genetic perturbations from context A to context B. So examples of context A to context B could be, cell line to primary cell, a resting state to stimulated state, let's say a mouse primary cell to a human primary cell, or patient sample from ancestry A to ancestry B. The idea would be in one of these samples to perform, again, TF-centric perturbation experiments like the ones proposed in the previous slide with multiple molecular and cellular phenotypes. In context B, you would measure a minimal set of steady-state molecular phenotypes and try to build models that can predict perturbation effects in context B by integrating the perturbation data in context A and the steady-state data in context B, and then validate this through perturbation experiments. I think this sort of design would distill, there are many interesting sort of versions of this, but this would distill into challenging problems that would actually expand the scope of these kinds of approaches. Problem two is a lack of methods that unify all types of variants. These are all the different types of variants, and typically we analyze these in silos. So there should be a call for datasets and approach that unify across all of these different definitions and through the lens of interconnected causal molecular networks. Problem three was a lack of evolving benchmarks and standards. I think there is a real need for setting up evolving community benchmarks and standards, evaluation metrics, and open challenges. This is a very serious problem currently in variant scoring approaches. We can go into this in detail, but that's one approach. And finally, for accelerating dissemination and discovery, I think there were interesting ideas on keeping interactive user interfaces and interactive analysis APIs as central focus, better data search and recommendation engines that allow us to discover data that's drowned in the deluge. And I'd like to also pitch for, just like we have data hubs and code hubs, I think models are going to be very interesting commodities to the extent that models should not just be sitting as accessory supplementary files corresponding to a paper, but we have to build centralized APIs around models for querying, scoring, and ranking hypotheses. I think that's the immediate sort of delivery you can get from these models. And then, of course, ideas for linking data hubs, software hubs, models used in publications, and improving data accessibility and sharing by removing and streamlining roadblocks. So I'll hand it over to David.